Good morning. Sorry for the late start. We had a few technical difficulties. My name is Tim Brewer. Welcome to the 6th Annual Consortium of Universities for Global Health Conference, Mobilizing Research for Global Health. I'm really delighted to welcome you here and to the city of Boston. A brief digression, as you know, Boston is a sports nutty town. And this past season started a little slow. They kind of took off in the middle there. And then as the season was coming to a close, it looked like Boston was going to fall short. There was some presumably questionable play calling on the part of Mother Nature. And in the final seconds, a snowfall did put Boston over the top for their record snowfall. Congratulations, Boston, on your snowfall record, but it is time to move on. Okay, so my job is to thank everybody who did all the actual work. And I would like to start with our host universities, Boston University, Tufts, the University of Massachusetts, and then the Harvard Global Health Institute, Massachusetts General Hospital. We're really delighted that they were willing to put in the time and the effort to allow us all to be here. I would also like to thank our sponsors. If it were not for them, we also would not be here. I'm not going to read through all the names, but I do want to highlight a couple. One is Child Family Health International, Jessica Everett, who does a tremendous job working with our students and trainees in global health. I particularly wanted to also acknowledge Boston University, who has gone above and beyond the call of duty in helping us to arrange and bring this conference here. The University of Washington Department of Global Health, which has supported CUGH from the very beginning, the Johns Hopkins School of Nursing and Dean Davison, who did tremendous amount of work to get us together, and then Lancet Infectious Diseases and the Annals of Global Health, which have been publishing our abstracts for the last 2 years, and we really greatly appreciate that, and all our sponsors for all that they have done. Of course, this requires people, and led by Dr. Gerald Kirsch, we had an extremely able and talented local organizing committee that put together the very exciting program that you're about to experience over the next couple of days. Our secretariat, these are the people in Washington. You may have gotten emails from them. Keith Martin, our executive director, will meet a little bit in a few minutes at the end of this opening session. Dalal Najjar, Karen Lamb, Jillian, Catherine, and Megan, all thank you very much. We couldn't have done this without you. Doris Steinbeck, who is running around somewhere, is the person who keeps us all together and keeps us all sane. Norman Stein also helped us to arrange this terrific facility, and thank you also to the Sheridan for everything that they've done. So I want to finally thank you. This would not be possible if you did not come. There will be over 1,600 of you here from 51 different countries, approximately 2/3 faculty and staff, and 1/3 students and trainees. That's really terrific. I'm very excited about meeting the almost 500 students and learning about the exciting things that they will be doing in global health and fixing all the problems that I was unable to take care of, which was pretty much everything. I would also like to thank our colleagues from all over the world who came, in particular the Asian Alliance for Global Health, the Canadian Council for Global Health Research, the Chinese Consortium for Global Health, the Latin American Alliance for Global Health, the Nigerian Academy of Sciences, South African Medical Research Council, the World Federation of Academic Institutions for Global Health. It's really tremendous that you can hear and share your insights and wisdom with us. I would like to ask 3 favors of you over the next 2.5 days. The first is that you get out of your intellectual comfort zone a little bit and go to a session that you would not otherwise go to because you know nothing about it or because you don't think it's relevant to what you do. Because A, I guarantee you will learn something, and B, I think you will be surprised at how interconnected the many varied things we do in global health are. The second is I'd like to ask you to get out of your personal comfort zone a little bit and introduce yourself to someone you don't know and ask them about what they do. We happen to have 1,600 of the most interesting people in global health hanging out here in this hotel. And if you get to know one or two people that you didn't know before, you might make a new partnership or a new colleague and spread your horizons. And then the final thing I'd like to ask you to do is when you go home, and particularly if you're working internationally, to invite a colleague to come to the meeting next year in San Francisco, because we all learn when we broaden our horizons and when we have a more diverse group of people together, and with your help, we can continue to build that diversity. So finally, that brings me to Dr. Gerald Kirsch. For those of you who have followed the stellar career of Jerry Kirsch as both the director of global and geographic and infectious diseases here at Tufts, and then the director of the Fogarty in Washington, now back here in Boston as like Grand Poobah for BU. It has a slightly more formal title than that, but I don't know quite what it is. You know, he's had a stellar career and he's really quite a supernova. We knew that when Jerry took this over, he would bring that same energy and enthusiasm to organizing this conference. Yes, you need sponsors. Yes, you need scientific program committee and you need organizers. But at the end of the day, you need one person with a vision and an energy to drive it all through. And that person was Jerry Kirsch. I was only peripherally involved, and yet, you know, Jerry sent me literally hundreds of emails around the planning of this conference, and that was just the first week. So please, I'm really excited about the next 2.5 days, and in large part that's because of Jerry Kirsch. Please join me in welcoming and thanking Dr. Gerald Kirsch.
Good morning, good morning. One of my jobs is always to correct him. And so the meeting's almost 3 days, not just 2.5 days. I would like to start off with the one slide that I have, because I would like to thank our local organizing committee, which represented the 5-member institutions in the Boston area out to the University of Massachusetts in Worcester. A superb group of people who were engaged and involved, and I thank them. I want to thank Keith and the Secretariat for everything that they've done, Norman for his sage advice, and Doris for doing everything that needed to be done. This is a 10th anniversary of the conception of CUGH. It was a meeting in Boston in 2005 at Boston University, which I hosted. There were 15 institutions represented, about 30 people in all. We had a little bit of support from the Borough's Welcome Fund. And the idea that we needed a consortium of the academic centers to take back the field of global health and put it where it really needed to be in our universities, and to be certain that we didn't over-medicalize. This was not about the health sciences. Global health is essentially about everything. And so rather than a professional school-based organization, We thought this needed to be at the universities, that it had to be inherently multidisciplinary, that it had to focus on the 3 pillars of academia, which is education, research, and service, and that there be some standards established for what is a good global health program so that our students come out of it with something valuable. Conception was followed by a rather elephantine gestation period, and it took several years before the delivery of CUGH. And thanks to Haile, Debas, and his colleagues at UCSF, we were established as an organization. And now we're infants. This is our 6th annual meeting since we became a formal organization. As Tim mentioned, we have had about a 20% increase in the attendance at this meeting. We bumped up against the fire laws of how many people we can register. And our committee decided that because global health must be informed by knowledge, and knowledge is generated through research, that we would emphasize and talk about how do we mobilize research to support global health. And once you do that, you realize, of course, that research is itself as inherently broad as the field of global health. It goes from upstream, maybe laboratory-based, to the downstream end of application and programmatic activities. And so we have tried to encompass all of that in the program. And yet global health is a very broad area. And so we've had a very interesting and exciting and thoughtful ongoing dialogue between our organizing committee and the Secretariat. We decided to do an experiment, and you are our guinea pigs. The experiment was, could we deepen our focus on a few things and still represent the breadth of global health? And the way we've gone about this is to pick 4 themes for the first 2 days of the meeting. And the 4 themes represent sort of a march from the upstream science to the downstream application, all focused on research issues. So we go from the technology revolution in genetics in our first plenary to the One Health agenda in the second, to non-communicable disease in the third, and to improvement science in the fourth of these. And then, in addition to the 2 plenary talks where we have 2 superb experts in each of these areas, we follow up with breakout sessions that also span from the upstream to the translational to the downstream. And I want to absolutely echo what Tim said. I had the same choice of words about getting out of your comfort zone. I would add, be bold. Go to something you know nothing about because you are guaranteed to learn. And science is really about how do we apply things from another field to what we're primarily interested in. So follow Tim's advice. I can't correct him on that. He's absolutely on target. We're going to ask you after the meeting to give us some feedback. We've tried to do a few inventive things in support of our students and trainees. We've organized a Meet the Speaker session where students and trainees were able to sign up to sit down in a small group with our plenary speakers, talk about careers, talk about the weather, talk about the Red Sox, whatever. We think this is going to be good, and we'd like to hear from you about that. We have, on the last day, those of you who are smart enough to be staying over through Saturday, we have 2 absolutely outstanding plenary panels. The first one was entitled Big Problems, Big Ideas. 7 of the global leaders will be spending a bit of time to briefly outline a big problem and then to think big out of the box of how we might come to solutions. And I think it's going to be extremely exciting. And then in the afternoon, we have a stellar panel on Ebola, and it'll really be focused on what are the lessons learned and what do we need to do and take action. So with that, I also want to add my welcome. Thank you for coming. The fact that we have an audience means I can sleep tonight, and I really appreciate that. And now it's my pleasure to introduce Haile Debas, who has been one of my great role models as a person, as a scientist. I just love him. Haile is here to help launch the first volume of the new Disease Control Priorities book that will be coming out over the next year or two. Dean Jameson, who's the editor, overall editor, is here. Haile is the editor of the first volume, which is on global surgery. He will make a brief presentation. And then in the breakout sessions after this first plenary panel, Haile is running one on global surgery. So I suggest the surgeons go to one of the genetics sessions and the rest of you go hear about surgery. Haile, thank you.
Thank you, Jerry, and I just want to also thank you for this incredible amount of work you've done to set this meeting. It's going to be very difficult for us in San Francisco to follow this. It gives me great pleasure to participate in the launch of the Essential Surgery volume of Disease Control Priorities, third edition, or DCP3. And I also want to thank CUGH for hosting the launch. DCP3 is a World Bank publication of considerable importance to policymakers and global health funders. It is a must reference book for all students and practitioners of global health. But significantly, the publication of this first volume, the Essential Surgery volume, signifies that creating access to basic surgical interventions is a smart investment for both population health and economic development. We've come a long way when a global health meeting begins with surgery. Do I have the slides? The first volume was published by the World Bank in 1993 as the World Development Report. It was influential in the corridors of policymakers and global funders. In 2006, the World Bank published the second edition of Disease Control Priorities, which featured a chapter in essential surgery. The full 9 volumes, third edition, Disease Control Priorities, will be published by the end of 2016 and will contain not merely one chapter as in DCP2, but a whole volume of essential surgery with 21 chapters. I know you're asking why surgery volume is being published first. To preserve the modesty of the surgical team for which we're so well known, I will leave the answer to that question to your imagination. The chief editor of all the 3 volumes has been Dean Jamieson, professor of health economics and global health, formerly at the University of Washington and now at UCSF. The DCP effort, in my opinion, represents a major contribution to global health. And the entire field of global health is indebted to Dean Jamieson. He has put on the global health map surgery, and the surgical professions are not merely grateful to him, but plan to make him an honorary surgeon. The 9-volume topics of DCP3 are listed here and span the whole spectrum of global health, from essential surgery to reproductive, maternal, newborn, child health, to cancer, mental, neurological, substance use disorders, to cardiovascular, renal, endocrine disorders, HIV/AIDS, STIs, TB, malaria, and finally, to improving health and reducing poverty. The 9 volumes of DCP3 share common elements. They all examine the estimate of burden of disease, deal with costing of essential packages, analyze intervention effectiveness, employ uniform economic evaluation, examine in detail and comment and recommend on policies, platforms, and packages, and discuss strategies towards universal health coverage. All 9 volumes of DCP3 also share one set of overall goals. They summarize and synthesize evidence of the effectiveness of comparative evaluation of global health interventions. They strengthen the capacity of evidence-based priority setting in global health. They introduce new methods for assessing the equity and financial protection considerations of health and policy. The 6 editions of Volume 1, the Essential Surgery Volume, the 6 editors of Volume 1 are listed here, and Charlie Mock and I serve as the lead editors. This is a whimsical depiction of the authors of the volume. We have 80 authors from 20 countries. 18 of the authors are from low and middle income countries. The volume is in 4 parts. Part 1 defines the health burden of disease requiring surgery using more rigorous estimation methods. It also estimates the burden of disease avertible by essential surgery. Part 2 deals with trauma, general and emergency surgery, obstetric surgery, congenital anomalies, hernias, hydrocele, as well as dentistry and cataract surgery. Part 3 deals with the surgical platforms and policies with emphasis on pre-hospital and emergency care, anesthesia, perioperative mortality, quality of care, and workforce innovations. and part 4, the last part, is devoted to the economics of surgery and examines in detail cost, cost effectiveness, and cost-benefit analyses. Here are the 5 key messages of the DCP3 volume on essential surgery. First, full provision of essential surgical procedures would avert 1.5 million deaths a year, or 6.7% of all avertible deaths in low and middle income countries. Essential surgical procedures rank among the most cost-effective of all health interventions. Third, effective and affordable measures such as task sharing have been shown to increase access to surgical care, while much needed investments are being made to expand capacity. Fourth, there are substantial disparities between countries of different economic levels in the safety of surgical care. Feasible and affordable measures such as the surgical safety checklist have been shown to improve safety and quality. And lastly, universal coverage of essential surgery should be publicly financed early in the path to universal health coverage, given that it is both affordable and highly cost-effective. Finally, how did I get there? Finally, DCP3 volume 1 is online and can be downloaded. We're also on Facebook and Twitter. You can also obtain more information by going to the University of Washington desk at the display. And of course, later on, there will be a panel by the editors on essential surgery. Thank you. Now gives me pleasure to introduce our next speaker, the Honorable Dr. Keith Martin, Executive Officer of CUGH. Keith.
That was loud. Good morning, everybody. It's a pleasure to be here, and thank you very much for being here. It's tough to follow Haile, but all of you should know that Haile and Jerry are 2 of the founders of CUGH, part of a small group that started this dream in 2008, and now look what we have today. So put your hands together, please, for Haile and Jerry. And Jerry and Tim did it. I actually also read those 100 emails a week when Jerry was firing them off. But I have to say that Jerry worked tirelessly for a long period of time to make this happen, and he sweated blood to make it happen. And Jerry, thank you very much. Now, also in my staff, there are a number of people who also work tirelessly, but none other than Karen Lamb. Is Karen here? Karen, put your hand up if you're here. She's very modest and probably won't put her hand up, but if you see her, thank her, because she's the conference manager and events coordinator for CUGH, and she is the one who works hours that are actually illegal. Maybe I shouldn't say that, but she does. And also Megan Adolphson, her conference assistant, has worked extremely hard, along with the rest of the staff. Dalal and Jillian and Catherine have all worked phenomenally to make this happen. So thank you very much to all of our staff. I only have a few minutes, so I have a couple of housekeeping issues that I think I'd like you to pay attention to, please. We have an app, CUGH 2015. Please check the Make Info page. You can download it on your smartphones. Please look at it because your CME credits are there if you're eligible to receive them. You can receive 16.75 credits and to complete them. But also, luckily, we also have dental credits. So for those of you who are dentists in the audience, you can also see page 29 of our brochure and take a look at that, and you can get your dental credits. I'd also like to thank, yesterday we had, as a measure of the success of our conference, we have satellite sessions the day before our conference. And yesterday we had a satellite session on global cancer with National Cancer Institute and Dana-Farber, doubling the numbers we've ever had before. There were also sessions with the Public Health Institute and also with the AAMC. And if the next year, if you can come to San Francisco, we will also have those satellite sessions. Also in your bag, please take a look because you'll see two journals. One, and we're indebted to both the Annals of Global Health and Phil Landrigan and Jack Narula, and The Lancet Global Health and Zoe Mullen and Richard Horton, because you'll see inside there both the oral and the poster abstracts that are going to be at this conference, so please check it out. So John Greenspan, who's here from UCSF in dentistry, asked, can I get the first slide, please? Thank you. So we were talking briefly about the conference and now what? Well, what I hope that we take from this conference is the ability to look at this slide and take a look at the opportunities that we have within this room and when we go home to take a look at what we have on your left, the power and capabilities we have within our institutions and our partners to affect what we see on the on your right-hand side, to prevent those tragedies and reduce that pain and suffering that all of us have seen in various parts of the world, including here in the United States. So we have unbelievable sessions during this conference that will do that. Please attend those if you can. CUGH has actually created a capability to do that that was born in part out of the Ebola crisis. If you take a look at your booklet and also on the app, we've created two things. One is a capacity building roster, a roster to use the capabilities that we have across disciplines to be able to build capacity across research, education, and service to implement and train in areas that don't have these capabilities. The other one is an emergency response roster. So we have the capability of actually bringing people and through another organization, deploying those people to meet HR needs in the case of a crisis. And so far, over 400 people have signed up to that. And thank you very much for those that have. If you haven't, please take a look at our conference app and also visit the CUGH booth where you can sign up for that. The other thing we have are interest groups across a broad range of global health challenges. We created them so to use as a platform for us to be able to work together to be able to address those challenges. And again, take a look at our CUGH website to do that. Lastly, we want to tell the world about what we're doing, right? So who here is on Twitter? Okay, we're using this hashtag CUGH 2015. And so I want to show the world what you're doing here. So I'm going to take your picture, okay? We'll do the official presidential picture, and then we're going to wave, okay? So let's tell the world who we are. Okay, now please wave. Fantastic. So you guys take orders well. Well done. So we're going to do this hashtag 2015. Please take the knowledge and information that you have here today that you're seeing, share it with the world. We're 1,635, but there are millions of people out there who want to know what you're doing and want to work with you. So let's collaborate with them through the Twitterverse. So now I have the distinct pleasure and honor of introducing the moderator of our first panel, Technology, Revolution, and Genetics. Dr. Robert Finberg is the chair of the Department of Medicine, the University of Massachusetts Medical School. I'll hand it over to him, and have a wonderful 3 days, everybody. Thank you.
Thank you very much, Keith. And thank you for all coming, and thank you to CUGH for having this session in Boston. I just want to say on behalf of the local organizing committee, which included people from Boston, Worcester, and other parts of Massachusetts, I want you to all recognize it's now spring, so I'm going to, at the count of 3, I want to have a cheer for spring, okay? 1, 2, 3, spring! It's actually here. So we have 2 outstanding people speaking at this plenary session, Dr. Venter and Dr. Werth. And I am going to introduce Dr. Venter first. He unfortunately is not able to share spring with us in beautiful Boston, but we are lucky to have him, and he is going to video conference with us from San Diego. Dr. J. Craig Venter is a biologist renowned for his contributions to the field of sequencing the first draft human genome in 2001, the first complete human diploid genome in 2007, and the construction of the first synthetic bacterial cell in 2010. He's the co-founder and CEO of Human Longevity Incorporated, HLI, a privately held genomics and cell-based therapeutics, diagnostics, and therapeutics company that's dedicated to extending the healthy, high-performance human lifespan. He's also the founder, chairman, and CEO of J. Craig Venter Institute and co-founder, chairman, and chief scientist of Synthetic Genomics, Inc., SGI. He's won numerous awards and accolades. He is the recipient of the 2008 National Medal of Science, a member of the National Academy of Sciences. He's the author of Life at the Speed of Light, From the Double Helix to the Dawn of the Digital Age in 2013, and A Life Decoded, My Genome, My Life in 2007. And hopefully, video and technology willing, we're going to have Dr. Venter, who's going to give his address, and then we'll have questions, and then after that we will go to Dr. Werth. Dr. Venter, are you here?
Good morning.
Morning.
I don't know if I show up visually there yet or not, but I'm pleased to at least be able to join you guys electronically. I don't know if you can see the first slide. Oh, there we are. Good morning. If I could have the first slide, please.
It's up to the side.
There we go. So that's the building I'm speaking to you from. That's the Monterey Institute here in La Jolla on the UCSD campus. It's overlooking the La Jolla Cove, the Pacific Ocean. The sun is just starting to rise, so that's what it looks like here. This building is the first carbon neutral research building in the world. The roof is a 0.5 megawatt power plant. And it's been a real challenge with a laboratory facility to get to carbon neutrality, but we're pretty proud of that. And I'm also reducing my carbon footprint by speaking to you electronically this morning. I'm going to talk to you about some of the latest things happening in our exploration of the genetic code. One of the key questions we've been working on for a very long time is, does DNA contain all the necessary information for cellular life? We've been taking a wide range of approaches to that. And as we try to do now thousands and potentially millions of human genomes, can we predict the phenotype directly from the genetic code? And then ultimately, can we rewrite that code? So this is the 20th anniversary of our sequencing of the first genome in history. And when we sequence a genome, we're converting the 4 letters of the genetic alphabet into 1s and 0s of the computer. I call this digitizing biology, and it's been happening at an ever-increasing pace. This is the science cover 20 years ago when we sequenced the first genome using some new mathematical approaches to assemble the DNA. 5 years later, we're able to use the same technology on a much grander scale for sequencing the first version of the human genome. This was a large undertaking. It cost about $100 million. Took my team 9 months to do it, but accumulated then the announcement with President Clinton at the White House and this paper in Science in early 2001. And then 6 years later, we published the first complete diploid genome. All of us have 2 sets of genomes, one we get from each of our parents, and sorting out that diploid nature is a very important part of understanding our biology. Now, 15 years later at human longevity, the scale is changing enormously. We now have these new machines from Illumina and PacBio. Each one of the Illumina machines is 1,350 times what Celera could do 15 years ago. So we now have 24 of these machines. So you can see how it's going to be possible to get up to hundreds of thousands of genomes a year towards our goal of 1 million genomes by 2020. This is exciting technology. Each of these boxes cost about $1 million each, but compared to a 4-story facility filled with 350 machines 15 years ago, the fact that each one of these is 1,350 times that gives you a sense of the change in technology over this 15 years. And instead of $100 million, we're down to about $1,500 per human genome. We're also using the amazing PacBio technology. This can read single molecules of DNA. It can read stretches in the tens of thousands of base pairs on a single molecule of DNA. And this helps give us a long-range continuity that, coupled with the Illumina short reads, allows us to put together some very accurate sequences by combining these 2 technologies. We're actually using the PacBio instrument to create on the order of 30 global reference standard genomes. These cost about $30,000 each versus the $1,500 for an Illumina genome. But right now, my genome is, as the first one sequenced, has been a reference. NIH has a sort of composite genome that would not live if it had to be booted up, but the number of references is very limited. And as we start to do population sequencing, the data from the Illumina machines does not allow for independent assembly of the genomes. So everything is layered on an existing sequence for comparison. So we want to get, so we have references from different ethnogeographic distributions so that we have appropriate references for all the different genomes we're trying to do. Our goal is to interpret the human genome. We want to be able to predict basically everything that's predictable from our genetic code. Right now, we're doing a clinical study. We have well over 500 participants already, where we're taking 3D photographs of their faces. These 3D photos allow us to do about 30,000 unique measurements on the faces. We take that into our machine learning algorithm, and we're developing a set of algorithms that can predict your photograph from your genetic code, which helps prove that genomes are in fact your ultimate identifier. You can't escape it. You can't de-identify the data. It helps philosophically to understand that, but it also helps with the identification in many cases. In the US, there's over 250,000 bone sets from missing children that have died that can't be identified. And simply from sequencing the DNA, we can generate a photograph of those individuals. We're going to be able to predict age from the genome. We have a new algorithm that can predict from your genetic code what the age is of that sample, disease risk, drug responses, etc. We've also tried to understand and interpret the genetic code. When you do a computational assay, you're looking at the ones and zeros in the computer. We wanted to see how much the data in the computer really represented life. So we went the other way. We started with the ones and zeros and rewrote the genetic code, starting with 4 bottles of chemicals. That's been our program in synthetic life. We started with a bacteriophage, PHX-174, that kills E. coli. We started with its code in the computer. We wrote out the genome for this organism and injected it into E. coli. The bacteria recognized this piece of synthetic DNA as normal DNA, started making the proteins. The proteins self-assembled to make the virus, and after a certain accumulation of viral particles, it lysed the cells and then it infected adjacent cells, and that's what you see here on this plate. The clear plaques are where the PHX has killed E. coli and has then spread. We call this a situation where the software builds its own hardware. We just put in a piece of genetic software and ended up with these infectious viral particles. Our goal wasn't to make small viruses. We wanted to construct an entire self-replicating bacterial cell. But we knew if we could make viral-sized pieces accurately, we could find a way to string things together to make an entire chromosome. And that's what we did. And it was a team that included and still includes the Nobel laureate Hamilton Smith, who discovered restriction enzymes, Clyde Hutchinson, who retired from UNC and has been working with us for a while. Dan Gibson is a young scientist who started as a postdoc and has grown to be a team leader in this space. You can see it took a number of steps, a number of great leaps technologically to go from where we could only make a small viral genome to where we could synthesize a 1.1 million base pair genome. So to build this, we started with making pieces that were 1,000 letters long. We put 10 of those together to make pieces that were 10,000 letters long. We put 10 of those together to make pieces that were 100,000 letters long. And we put these 11 100,000 base pair synthetic DNA pieces into yeast, and it was self-assembled into the complete chromosome. We isolated the chromosome from yeast, and with the technique of genome transplantation, booted that up in a cell. So genome transplantation is a process that took us quite a while to develop. 2 scientists responsible for that were John Glass, who's the team leader here at the Institute, and postdoc Carol Latique from France, put in unbelievable hours to get this to work. But essentially, we can insert a chromosome into a recipient cell. So that's what we did with the synthetic chromosome, as you can see in this sophisticated movie. When we insert the chromosome into the cell, we now have a cell of a given phenotype, but it has 2 sets of genetic instructions. What we think happened as soon as we put the chromosome in the cell, its genetic code started to be read immediately. Proteins were produced, and some of the early proteins produced are the restriction enzymes that recognize the chromosome in the cell as foreign DNA and shoot it up. So now we have the phenotype, the body of one species, and the genetic software of another. So what happened? In a very short time, we ended up with these gorgeous blue cells. And when we interrogated them The only DNA in the cell was the synthetic chromosome that we started with. And every single molecule in the cell derived from that synthetic chromosome. There wasn't a single molecule of any kind left from the original species, the original cell. One way we can tell synthetic DNA from natural DNA is we can watermark it. This was the issue of Science in 2010 where we published this initial data. We have the ability to write the entire English language with numbers and punctuation just using the genetic code. And so one of the tenets of synthetic biology is to watermark the DNA so it can't get confused with natural evolution. And these watermarks can be quite sophisticated. We had instructions in there to, if you decoded this information, there's a URL embedded in there so that you could actually send an email to this species saying that you had been able to read its genetic code with the names of the 46 scientists that contributed to the work, the institutions, and I added 3 quotations from the literature to help celebrate the event. The first was from James Joyce, to live, to err, to fall, to triumph, to recreate life out of life. The second is from Oppenheimer's biography, American Prometheus, see things not as they are, but as they might be. And the third is from Richard Feynman, what I cannot build, I cannot understand. When this was widely publicized, one of the first responses we got was a letter from James Joyce, a state attorney, asking us if we had copyright permission to use this quotation. And obviously under US law, you can use up to a paragraph or so with attribution, but without permission. And then we got letters from a Caltech scientist saying we had misquoted Richard Feynman, and they finally sent us a photo of Feynman's blackboard from the Caltech archives showing what the original quotation was. And we think his biographer might have actually changed the quotation. So the original one was, what I cannot create, I do not understand. I think that's much better than what ended up in the literature. So we've gone back to change the genetic code so Richard Feynman can also rest happily. We think all this data helps prove that life is a DNA software system. And if you change that software, you change the species. So this has lots of implications, including our basic understanding of life, but it also allows us to try a lot of new applications to science and medicine using these new tools. We have computer software for helping us to design DNA software, and we've been working since 2010 to try and design a cell from scratch in the computer. It's been a huge challenge, in part because about 10% of the genes in our final construct are of unknown function. Our basic knowledge of biology is so limited that 10% of genes in the smallest organism have yet to have their functions determined. So when we get to the human genome, it's much of a bigger fraction and much more complicated. We actually have a design, build, test cycle for synthetic DNA, synthetic cells, that we have to go through these iterations because of the unknowns that you can't just design in a priori. I was giving a lecture up in Seattle where my late uncle attended the lecture, and he was a Boeing designer that designed the 757. And I said, imagine if in designing Boeing aircraft, the designers didn't know what 10% of the components did. His response was, what makes you think we knew? And you've seen the cycles of aviation where a failure is examined thoroughly, tried to be corrected in the next iteration, so the planes won't crash. And that's basically the cycle that we use with designing genomes. So in 2010, our first version was synthetic genome 1.0. We've been trying to minimize that through direct design and then trial and error. And then for future design, we've been trying to do what we call defragmentation of the genome, undoing the messiness of 4 billion years of evolution to design things by cassette. So with natural evolutionary processes, events are very random. Things get inserted all around the genome with no logic to it at all. With our defragmentation, we're actually organizing all the processes by function. And the amazing thing is this actually works. We can separate everything in classes and groups and have it be highly organized, but this actually takes a lot of effort. This is just a 1/8 segment showing all the changes in the genome going from the natural genome to the reduced and reorganized defragged genome. This is important for design. So if we're designing organisms, we can just pop in cassettes for translation, for what type of energy, et cetera, that we want to use. It also leads us to the ability to do a variety of things that weren't possible before. We've been working on algae cells for some time, both as an energy source and as a food source. And it's very difficult to change the metabolism of algae cells, in part because eukaryotic algae cells are actually a combination of two species. So the entire plant world and all the eukaryotic algae are a result of a photosynthetic bacteria being inserted and maintained inside the eukaryotic cell. And so these processes are very compartmentalized in the cell, but we found we can actually create and add an entirely new chromosome to the cytoplasm of the algae cells, changing their fundamental biology and the processing of molecules. And with this, we've succeeded for the first time in changing the energy levels of the cell. So another example of changing the software and changing the species is a project we're working on at Synthetic Genomics with United Therapeutics. We're trying to rewrite the pig genome to create humanized pig organs for transplantation. There's close to 1 million people in the US that die each year from the lack of organs for transplantation. And so we are trying to change that by improving the ability to use allograft transplants. And pigs have been considered for some time to be good sources of possible replacement human organs because of the right size and function. For example, the pig heart is almost identical in size and function to the human heart. And so by trying to change the genetic code, we're trying to get it so we can deal with these very complex rejection responses. Some of them take place in minutes. Long-term rejection is over days to weeks. So with xenotransplantation, for it to work effectively, we have to deal with all these different processes. And we have a complex setup where the team at Synthetic Genomics is working on the genome design and genome changes. And our goal is to deliver to the United Therapeutics team a totally new fibroblast cell line with humanized pig genes in it. The United Therapeutics team then does nuclear transplantation to take the part human, part pig genome and insert that in cells to create living pigs from it that are then grown up and then the organs tested for transplantation. So you can see it's a complex cycle. It's a wide range of important skill sets involved in this, but some of the early results are very exciting and very promising for this key unmet medical need. And to do this, we're having to create all kinds of unique genetic tools to be able to insert things in the pig genome without disrupting a normal function. So it's an exciting stage. It should be obvious by now that if we can go from reading the genetic code into the computer and then go the other way, we can actually interconvert the digital world and the biological world. And we do this in one process that we call biological teleportation, where we can actually send biology through the internet. We've been working on sending units and receiving units. The sending unit we've been testing with NASA at the Mars test site in the Mojave Desert, where we actually sequence samples isolated from the desert. It's immediately on-site beamed up to the cloud, and then back at the Institute where we're building a receiving unit with synthetic genomics. We take that digital information and convert it back into biology. So we've been doing this with funding from DARPA. What you see here is the prototype machine that all it needs is a digital signal, and it can make whatever comes in through that digital signal. So we've had a variety of rigorous tests from DARPA. We've had to make DNA, we've had to make RNA, we've had to make proteins. So we now have, with this device, the ability to download proteins such as insulin from the internet. We had to produce a phage. We had to produce the flu vaccine and a number of functional RNA molecules. So we've proven that all of these now can just be sent digitally through the internet and downloaded. And the implications of this obviously are huge. We've been using this for global vaccination, and I'll talk about that in a minute. We have this technology, miniaturized version of that early prototypical device that does part of the effort. It actually assembles large pieces of DNA automatically for people. This is the first biological instrument that has had multiple security levels built into it. So with the potential misuse of this technology, we're trying to keep as much security control as we can, so somebody doesn't make an infectious agent from this. And so it has a large number of security levels built in, hopefully to prevent that. We applied this first to making new flu vaccines very rapidly. This has been successful now, starting with a real-world test with H7N9. My institute is part of a global flu emerging virus sequencing effort, tracking new emerging strains from around the world. When the first outbreak of H7N9 occurred in China, the samples were quickly sequenced and posted to the cloud. We downloaded that digital information and within a week had produced a new vaccine candidate that went into production. And for quite a while, our synthetic H7N9 virus was the only source in the US for the CDC and vaccine development because the difficulty in getting the actual strains out of China and shipped around the world. Novartis has North Carolina, a cell line based facility instead of using chicken eggs. We have one of our devices there, so with any new flu strain, we can send the information there in a fraction of a second. And there exists the potential for making multiple vaccines per year. But as you've seen with this last year, the vaccine that's out there doesn't match the circulating strain because we've been using ancient technologies for making these decisions. If we use these new technologies, even if the strain changes during the year, in a short time, a new version of the vaccine can be reproduced. We can also, by miniaturizing these facilities, be able to stop any pandemic before it starts. Because just with one of these units, you could rapidly produce vaccines right on the spot by sending the information digitally. With the H1N1 pandemic, it was until several months after the peak of the pandemic that the vaccine was available. It took almost 9 months from the start before a vaccine was available. Now we can do that in a matter of weeks. And the potential exists for just having one of these devices attached to your own computer so that you can download insulin or download other biologicals. And a new flu vaccine could be sent around the world in a fraction of a second. If you have one of these devices, you could download it and we could prevent any future flu pandemics. We're trying to develop similar things for antibiotic resistance. We've been using phage, and these are turning out, when applied properly, to be extremely effective. These are the viruses that attack and kill bacteria, as I showed originally with CUGH. And we've been engineering a new set of viruses, expanding the range. For example, trying to kill Pseudomonas and burn infections on the skin instead of having a huge cocktail of dozens and dozens of phage. We've changed the host range by just genetically engineering the phage to kill more strains of Pseudomonas. And this is turning out to be very effective, and we've just submitted this data for publication, and we think this is the first actual engineering of phage to change their host range. We're trying to take this comprehensive integrative approach to medicine, particularly at HLI, where we're not just dealing with genome data, we're really focusing on phenotype data, clinical information. We're measuring the microbiome, we're measuring 2,400 chemicals in the bloodstream, and in some cases measuring proteins. We're combining all this data in a new comprehensive machine learning approach with integrated health records. I'm sure you all know about the microbiome. The first study of this was done at the Venter Institute with Karen Nelson, who's now the president of the Institute. We have roughly 100 trillion human cells. You have 200 trillion bacterial cells. And these are a very important part of physiology, resistance to disease, even causes of disease. So we're sequencing the microbiome along with the human genome. And as I said, we're measuring these 2,400 chemicals in the bloodstream with Metabolism in North Carolina. And in our own phenotyping centers that we're opening later this year, we'll be doing the most comprehensive assessment of somebody's physiology, everything from the human genome to quantitative brain and body MRI imaging, and measuring all these other functions. Using new MRI imaging techniques, we think it's now possible with the algorithms developed by a team led by Anders Dale at UCSD. They can convert these complex images of several gigabytes of data into files comprehensively measuring the volume of different brain regions that we can compare back to the genetic code. And our goal is to eventually be able to predict the brain structures straight from the human genome and understand disease risk from the earliest stages. This data is already starting to transform cancer therapeutics. Pfizer thought they had a drug that could be used for treating lung cancer. They did a large clinical trial and it badly failed. But when it was found that there's a gene called ALK, when there's a translocation in that gene, about 4% of lung cancer patients have that translocation. And if you're a lung cancer patient, you have that translocation, the Pfizer drug is effective in more than 60% of patients of greatly reducing the tumor burden. So this is now part of the definition of precision medicine that President Obama recently announced. So we're pleased to see the government is now catching up with this. Genentech has been doing this with Herceptin with breast cancer. And now it's gotten to, if you have cancer, the most important thing you can do is get the genome analysis done on those tumors to understand the driver mutations, which determines which therapeutics are used. We're integrating this data to try and make sense out of it. I hired Franz Ock out of Google. He built the Google Translate system, which is now up to 90 languages using machine learning. And now he's got a team that's applying that to all this complex data to make discoveries and predictions from the human genome. And obviously integrated data is better. We're reducing the complexity of the data for these huge calculations. And we're using multitask computational learning instead of asking linear questions, which is all that's been possible in the past. We're trying to convert these massive amounts of data into new knowledge. We're trying to set it up so anything that we can precisely measure in the human condition, if it has a genetic component, we will be able to discover that and measure it. We're working with multiple centers to collect unique disease cohorts. We're sequencing those and combining it with their clinical and phenotype data. And we're expanding this to direct healthcare hubs that will be the first to comprehensively measure this data and use it in turn for an entirely new approach to medicine. Thank you very much.
Thank you, Dr. Venter. And it was really an exciting tour of all the things that are going on there. We do have time for a couple of questions, but I want to give Dr. Werth time as well. So I will ask people in the audience to take a microphone quickly to ask Dr. Venter any questions about his exciting talk. Questions from the audience? Hard to believe there are no questions. Tell us a little bit about your view of the controversy, because you mentioned it briefly in terms of release of genomic information, which has now become a huge issue. There's an editorial about it in the New England Journal of Medicine just recently. Can you tell us some of your feelings about protection of human genetic information and what the issues involved are?
Well, to us, in what we're doing in human longevity, we consider it absolutely essential that this data be in highly secure databases. The federal rules still have a silly notion associated with them that you can de-identify human genomic DNA as long as you don't have the person's name or social security number or other identifying material such as a photograph accompanying the clinical records in the genome. This is really quite silly because, as any trial attorney can tell you, your genome is your ultimate identifier and does everything from getting people out of prison to putting people in prison if they can be associated with DNA at a crime scene. The fact that we can predict your photograph from your genetic code means it can never be de-identified. And so we consider it's very important unless people voluntarily do something different, that the data should be in highly secure databases. And that's how we're treating the data. We think all the clinical records combined with the genome should be all in HIPAA compliant databases. My genome is on the internet after it being the first one sequenced. I felt it was key to try and demystify this field by having data available. It's not something I recommend to most people. And knowing what we know now about the genome 15 years later, I might have reconsidered that decision. So I think the security of the data is very important for people's protection, but these databases are going to be so valuable for preventative medicine, for major new discoveries, that it's an exciting phase for the future.
Okay, great. There's a question. I'm going to take one in the back there.
Dr. Venter, can you hear me?
Yes.
Yeah, hi, it's Glenn Galton from the University of Pennsylvania. Since you're thinking out of the box, which you're well known for this morning, and you talked about digitizing the infectious organism, for example, and developing new vaccines, but you know, a limiting factor in that, as you referred to, is the construction, essentially, in the case of flu, of the delivery cell vectors. How far are we from thinking about say, a molecular version of like three-dimensional printers so that you could actually get the information and print the vaccines on site in global locations?
Well, that's essentially what I was describing. Our digital biological converter, in fact, is the equivalent of a 3D printer. The information we send is a digitized set of the DNA that we convert back into the chemical DNA, which could be, that same box can actually convert and hand out a vaccine that could be injected on site. So, as I said, you could we could miniaturize this so you could have a home version of this attached to your computer, but right now it wouldn't be that hard to replicate the giant facility that Novartis has in North Carolina to make smaller versions of it distributed around the world and eliminating any future flu pandemics.
I'll take one very brief question, then we need to move on to Dr. Werth.
A lot of work going on epigenomics. What is your experience? How do these epigenomics will affect on the primary genomics? You have any experience on that?
Consider epigenetic phenomenon as different from genetics. So our data with the synthetic genome has shown that everything in the cell, including epigenetic phenomenon, are coded in the genome directly. So, but there are important modifiers. DNA methylation of the human genome affects, in part, whether your mother's chromosome and genes are more prevalently used than your father's. So it's important to understand it as well on scale.
Okay, thank you very much, Dr. Venter. We really appreciate your coming here and very much enjoyed the talk.
Thank you very much.
Okay, our next speaker is Dr. Diane Werth. Dr. Werth received her PhD from MIT. She is currently the Richard Pearson Strong Professor of Infectious Disease, the chair of the Department of Immunology and Infectious Disease, School of Public Health. In addition to that, Dr. Werth is a senior associate member of the Broad Institute, co-director of the Infectious Disease Initiative at the Broad Institute, the director of the Harvard Malaria Initiative, and she's the faculty chair of the Harvard Integrated Life Sciences Graduate School of the Arts and Sciences. She's a member of the Institute of Medicine and has made major contributions in the areas of malaria research and pathogenesis and study of many infectious diseases across the globe. And she's going to talk to us about harnessing genomics for global health. Thank you, Dr. Werth.
Great, thank you. Well, thanks to the organizers for the invitation to talk about this topic. And really, I think Craig Venter has really set this up well. He's really talked about, thinking outside the box, looking to the future. In fact, what I'm going to talk about today is using genomics to understand what's happening in the natural world. And I'm going to focus on the pathogens. And there are two reasons to focus on the pathogens. One, of course, these are the organisms that end up causing many of the diseases that fall into the categories of global health. But also, from a genomic standpoint, these are much smaller, much more rapidly replicating organisms, and therefore you can actually see evolution in real time and begin to understand how the forces of natural selection or artificial selection are beginning to change the organisms that we interact with every day. My own work has really been focused on malaria, but I have, for this, decided to also bring in some work of others, which I think exemplify some of the applications of the genomic principles and the principles of evolution to understanding infectious diseases. Okay, so let's see if this works. Okay, so it's basically this Red Queen dynamics, and that is there's an arms race between organisms that infect humans and the human host. And it is that survival of both that leads to this, sort of epistatic interaction, this interaction which allows both organisms to survive, and fitness of either the microorganism or the human host, or in the case of vector-borne diseases, the vector plays a major role in this interaction. And that's really what we're looking at when we look at natural selection. Okay, so we know this is actually an op-ed piece with many of you are familiar with. This is in 2003 at the time of the SARS epidemic. This is just the size of the banners. This was an op-ed piece in the New York Times. The size of the banners reflects approximately the burden of disease in 2003. And of course, one of the points of this, I think SARS is down in the lower right-hand corner. I don't have I do have a pointer, but I'm not sure how effective it's going to be. SARS is down here in the right-hand corner, indicating in terms of global burden, it wasn't very important, but it certainly created enormous interest. Okay, but since then, we've had others. Ebola, you've already heard about today, and there's going to be a special session. Chikungunya virus is another one of these emerging infections, and I'm going to come back both to Ebola and to chikungunya during the latter part of the talk, just to give you examples of how using genomics you can begin to understand how these organisms, these viruses, are evolving. Okay, as Craig has already told you, the reason we can do this is because of the vast changes in the technologies associated with genomic sequencing. The first malaria genome cost about $20 million. It now costs less than $100 to sequence a malaria genome, and in fact, the major cost is extracting the DNA and preparing it for sequencing. The actual sequencing costs are probably in the sub $10. So this has really changed what we can do. And also the technology, and Craig gave a sort of much sort of more expanded and future-looking version of this, but even today, we can essentially take a sample from a patient in the field and be able to identify both the patient's DNA and the organism's DNA, and then be able to use that both to understand the sequence, to define markers. I'll talk a little bit about defining markers for drug resistance or other kinds of changes which might be useful in surveillance. And so we have a whole pipeline of methodologies that we can use to be able to not only find out the information, but translate it into practical applications. Okay, as I think about where we are today in using genomics to really advance the area of global health, and in particular the area of infectious diseases, the major advances are really in the areas of surveillance, understanding transmission and spread, the detection of new traits, whether that's in the case of drug resistance, a very important issue in many infectious diseases, including malaria, which I'll talk about. Whether it's expanded host or vector range, this is going to be an example of Chikungunya virus, where a single change in the virus has changed the ability of the virus to grow in different, or to be carried by different mosquito species, greatly changing the range of the transmission of this disease. Or in fact, looking forward, as Craig was talking about vaccination, vaccination is in some ways a very specific kind of drug, and in organisms where antigenic variation, where the changes in the organism occur in real time, as we see with influenza, as I'll tell you we see with malaria, in fact, we are going to be able to use genomics to understand how vaccines are working or not working. I also think another area, which I won't spend a lot of time on, but I think is an area where we are just beginning to see the value of genomics in the area of innovation and discovery. I'm going to give you some very specific examples from malaria, where we're using genomics to identify new drug and vaccine targets and new targets for insecticides. And finally, I think that one of the very important things, and particularly at this meeting, is this notion of education and leadership development. I think one of the exciting things about genomics, and I think Craig referred to this, is in fact it's just information that can be reduced to its most essential level, and that really reduces the barriers for being able to work with it. And I think education and leadership development worldwide is something which has been a key focus of our programs, and I think is going to be a key to actually changing the dynamics of how discoveries are made in many areas, but in this particular area of infectious diseases. Okay, so I'm going to start with malaria. As many of you know, there's been this challenge put down by Bill and Melinda Gates in 2007 to eradicate the idea of eliminating and then eradicating malaria. And for those of us who are biologists, I think this is a daunting task, but I think it's something that caused a paradigm shift in thinking, and I want to sort of go through that with you, looking at the genome lens. So in some ways, this is a way that at least I think about how one might begin to imagine eliminating an organism. This is sort of a natural history of Plasmodium falciparum. It started as a primate malaria, and its closest ancestor is a primate malaria. Then the parasite eventually got into humans. It probably took multiple steps, but then eventually an organism evolved that was able to grow effectively in humans and be transmitted from human to mosquito to human. That led to a worldwide expansion of the parasite, and then as this happened in Africa, as people migrated out of Africa, as humans migrated out of Africa, the parasite migrated with them, but there was this founder effect. And then, as we think about it in the modern times, the parasite, actually all of the parasites existing today are the survivors of these multiple bottlenecks or selections on the parasite. And we see footprints of this. In the 1950s, malaria was the target of elimination and eradication, and what came out of that was highly chloroquine-resistant parasites because the drug was used extensively and the parasite evolved resistance. So again, today, starting in 2007, there's been this massive step up of efforts to control or to eliminate the parasite from humans and from the mosquito vector. And that's putting enormous natural selection on the population. And you can imagine this is the plan. The plan is, this is from Al McGill's presentation at ASTMH this year, where in fact the idea is to use the existing tools and develop new tools to reduce transmission. Okay, and in fact, that's successful. This is just showing, comparing transmission in 2 time frames in 2000 versus 2010. And you can see, and I'm going to talk about some of our own work in Senegal, where we actually, the country has seen an enormous reduction in the number of reported cases of malaria. And so this works. You can create, or you can select, or you can begin to eliminate organisms. But of course, that has the effect of potentially changing or selecting different organisms in the population. And let's see if I can make this work. And we're beginning to see that. Now, I'm just going to digress for a moment to tell you about a set of tools we've developed to be able to essentially mark or barcode every single parasite in the world. And the practical application of this is, sequencing full genomes, although doable and not terribly expensive, turns out not to be necessary to actually rapidly identify parasite biology. And so we've created a code. This is a molecular barcode. And we've used that to begin to interrogate parasites in Senegal, after this introduction of these measures to reduce the parasite burden in the population, both in humans and in mosquitoes. And this is just a series of years. And what we're showing here is, in these colored bars, is the increase in parasites that are highly related to one another. So this is what we're seeing is a bottlenecking, a reduction in the population of parasites. So this tells us that, in fact, the intervention is working, and we're seeing it at a genetic level. We can, in fact, map and find the related parasites. So we can actually map which parasites are related to one another. They've been transmitted together. And this gives us enormous ability to begin to map transmission trees and understand what is happening at the sort of individual level in order to better modify treatments. However, in collecting this data, we began to realize that we needed the assistance of mathematical modeling. We collaborated with the Institute of Disease Modeling in Seattle, headed by Phil Eckhoff. And interestingly, they took our data and began modeling what was happening in terms of transmission. And indeed, what they saw is at the introduction of these measures, you begin to see a reduced transmission, but then the best fit in the model was, in fact, a rebound. When we went back and looked in detail at the data, although it wasn't picked up at the level of the ministry, in fact, there was this reduction, but then what looked like noise turned out, in fact, to be a rebound. And this is one of the advantages of having these kinds of surveillance tools. One can, in advance and in real time, begin to anticipate these kinds of changes in the population. And I think this is extremely important as we go forward to understand what's going on as we begin to move to disease elimination and eradication. And you'll see that in the other two examples. So the idea is to actually use genomics as essentially a thermometer to determine the level of transmission in a very straightforward, high throughput way, which can readily be done in country. This entire technology and most of this data was collected in Senegal by the team there. Okay, but of course, why do we see this rebound? Well, there could be many reasons, but certainly one of them that we see worldwide, not necessarily yet in Senegal, is the emergence of drug resistance. And this is a major problem in almost all infectious diseases for which there are effective drugs. Here I'm just depicting resistance to the artemisinin derivatives in Southeast Asia. This is now a focus of an focused elimination program to get rid of artemisinin resistance in Southeast Asia. And I'll come back, we'll come back to the insect vector. This is pyrimethamine resistance, or resistance to insecticide, is an equal problem, in this case in Africa. In the case of artemisinin resistance, work by a number of groups led by the Institut Pasteur has identified a single mutation or a group of mutations in a single gene, the Keusch protein, that seem to be associated with this reduced susceptibility in the field. And now that's being used as a marker to look for drug resistance in other parts of the world and to understand the flow of drug resistance throughout Southeast Asia. Again, this falls into the surveillance category, but it has very important potential practical implications. Okay, so we'll come back to malaria in a moment, but now I want to turn to Ebola and Chikungunya virus just for a few minutes to see that these same kinds of approaches can be applied to understand the epidemics and their transmission. We all know about Ebola virus. This is work of Pardis Sabeti's group with their collaborators in Sierra Leone. The Ebola virus spread very rapidly. Pardis's group was on the ground and preparing actually for a study on loss of fever, but was there and able to be prepared and able to do the first molecular diagnosis in Sierra Leone of the Ebola virus, and then very rapidly collect samples from these initial cases. They were rapidly processed, as Craig described, and then the sequences obtained. And they were able to identify, and I don't know how well you're going to see this, but this is the 2014 virus, and these are the viruses of previous Ebola outbreaks. So it's clearly a very different virus by genetic distance. And in fact, if you look at the outbreak in Sierra Leone, you find that it's derived from Guinea, but then expanded and was beginning to mutate in Sierra Leone. And this kind of work can map exact transmission trees. So again, very important both surveillance, but also transmission and spread to understand how the disease is spreading using these unique molecular markers. Obviously, it's important both to have the infrastructure and the trained individuals on site and ready to do this, and I think that's a very important theme, which I'll come back to a couple of times. And it's also important, I think, to realize that this is an iterative process, that in fact, knowledge from the genomics can feed into developments of new vaccines or drugs, can lead to interesting conclusions in terms of the spread of disease and important implications for public health. But it's also important that data from the lab go back to the field to inform what's going on in the field and then to watch as this evolution is occurring in real time. In the Ebola case, that's evolution over a period of a few months. In the case of the emergence of drug resistance of malaria, that's probably a 5 to 10 year emergence. So these are in real time, and they're things that are important for decision makers. I also want to talk briefly about Chikungunya virus. This is work of Scott Weaver and his group and a number of people in this area. But Chikungunya virus is one of the, I would guess, emerging now or emergent infections, which has now a nearly worldwide distribution. And I think of particular interest in the United States, there's the potential that this will now spread quite rapidly into the southern United States. This is a virus like Ebola that has an animal reservoir. The virus can get into humans near where humans are near the animal reservoir and bitten by the same mosquitoes. The important transition of this virus is when it goes into urban areas, but probably most critically is when it changes from primarily being transmitted by Aedes aegypti to being transmitted by Aedes albopicus. And that mosquito has a much broader range, and therefore the disease can spread much more broadly. And so here is just the spread of Chikungunya virus. This was an animated slide from Scott, but I decided in terms of time I would just show you this to realize that, again, this is a virus that's spreading worldwide, reflective of transportation and business interactions, and is now in most parts of the world. When it was sequenced, it was clear that there was an adaptation in a single protein. This is the viral coat protein. There's a single amino acid change which allows this virus to grow both in Aedes aegypti and Aedes albopicus. So one single change in this 10,000 base pair genome leads to this change in host range. And now, of course, it's possible to follow this. Now that we know about this change, one can survey for this, and perhaps this is it gives us insight into developing new interventions. Okay. So that's what all I'm going to say about surveillance and understanding transmission. I think the tools are incredibly useful, and the methodologies for using them and getting data in real time exist today and are being used. I think the other important thing to understand is that having genomes available and having the ability to use innovation to discover new things. Craig talked about synthetic biology. I'm going to talk about more kind of in vitro selection. So it's not really synthetic biology, but I guess it's synthetic selection. And so this is probably, this happens naturally, right? If you test, if you have a group of organisms and they carry different genes, you can test for the correlation of genotypes and phenotypes. If you think about drug resistance, if this is the drug resistance gene, if you have a mixture of cells in a test tube, or in humans or in a population, if you exert a selection pressure, then basically you select this gene and all of its adjacent DNA to lead to the phenotype that you're selecting for. So this happens, I've shown you examples of malaria resistance, in artemisinin resistance in Southeast Asia, the change in the Chikungunya virus, which leads is selection based on mosquito host. But it turns out we can do this in the laboratory. And so it actually allows us to begin to identify new potential targets for developing drugs. As many of you know, there's been this vast effort in the areas, particularly of malaria, but other tropical neglected diseases, to screen chemical libraries for new chemical entities that kill the organism. But the problem with that approach is you end up with a chemical entity, but you have no idea what the target is. And so this allows you actually to identify targets, and it also allows you to look at other things which probably relate to how well this kind of resistance will survive in nature. I won't have time to talk about that, but just to give you a sense. So the way we do this is simply to grow the parasites in culture and then select with the agent that kills the parasite. And you do this repeatedly, and you come up with resistant parasites. And because of the genome technology, you start with a clone, you have the mutant, you start with a clone, you have the mutant, and then you are able to simply select, sequence, and compare the sequences of these organisms and find the target. And this is amazingly successful, not only in malaria, but in several other organisms. And I think it's the way going forward. So here is just, this is a halofuginone. This is a natural, the derivative of a natural product, febrifugine, from the Chinese pharmacopoeia. It's been used for years in treating Eimeria, but no one ever knew the target. We found it by simply selecting Plasmodium falciparum. It is the proallele tRNA synthase that contains the mutation. This is the enzyme that transfers, this is the enzyme that transfers proline onto the proallele tRNA, and it is an essential enzyme for the survival of the parasite. And our hypothesis is this is the target. The mutation, that we actually isolated 2 separate mutants in the same gene, in the same amino acid, but different changes. And there's lots of interesting biology. But this is now a tool to allow us to be able to identify the target of any small molecule that kills an organism. Again, an extremely powerful tool for discovery. We've now done this. We're part of a consortium funded by the Gates Foundation of 5 labs, doing this for essentially all of the high priority new small molecules. And we've identified a number of new targets as a group. Okay. So I think that's the summary of that. I think the final thing I want to talk about in the discovery side is, let's not forget the mosquito. Of course, we know that resistance to the pyrethroids is a significant problem for the future of our ability to control malaria transmission, and this just shows a timeline of occurrence of the pyrethroid resistances. And in fact, I think one of the very important things to begin to think about, to begin to think outside the box, and the genomes of these organisms are giving us the ability to do that. Essentially, currently, all insecticides essentially kill mosquitoes, all insecticides in use for medical purposes. And if a mosquito is resistant, it's able actually to survive and then expand, and there's a very strong selection. One of the strategies that Flaminia Cataraccia, who's in our department at the School of Public Health, is doing, is trying to think about ways to have resistant or have insecticides that focus on sterilizing the mosquito, with the idea that any mosquito that's resistant would no longer be able to reproduce. Again, thinking outside the box. And the genome sequences now of the major Anopheline species that transmit malaria are now fully sequenced, and we're beginning to get ideas from the genome about how to target the mosquito vector. And mosquitoes, of course, are also complex biological systems, having their own biology, a very active microbiome, and then interactions with not only mosquitoes, not only parasites, but other infectious agents. And so again, an area of enormous opportunity. And finally, just to make sure that we keep this interesting, the other very important area that I think we've just begun to scratch the surface is this interaction of intracellular pathogens, particularly complex pathogens like eukaryotic pathogens, the parasites, with their host cells. And in fact, this is from Barbara Burley's work in T. cruzi, but the concept is that the parasite actually kind of plugs in to the host and uses the host biology to be able to fully adapt its life cycle. And this is an area we're just beginning to understand the selective forces. But I think this study, the ability to study large-scale natural situations is very important. Finally, I think key to translating the knowledge that this technology is going to offer is in the area of education and leadership training. We're actively involved in that in many ways, very important for both the developing world. This is a training seminar we've done recently in Dakar, Senegal. The course that we offer on the science of eradication. And finally, I think obviously this data has to be taken into consideration in the broader sense of integrating the knowledge that we get from genomics with all of the other factors that play a role in the response of individuals and populations to infectious diseases. And with that, I'll stop. Thank you very much.
Okay, I appreciate that. Thank you for this another view of the parasites world. I think we do have a networking break, so maybe we should stop at this point to continue the conference to go on ahead. I think Dr. Werth will be around. Thanks very much.
Thank you.